Hello and welcome to pH Biology. Today's explainer is the enzyme, a biological catalyst that speeds up the chemical reactions of life. By the end of this video, you'll know what enzymes are made of, why they're specific, and the 3 digestive enzymes you must know cold. An enzyme is a biological catalyst. It speeds up a chemical reaction without being used up. Enzymes are proteins with a specific 3-dimensional shape that includes an active site where the substrate binds. Each enzyme catalyzes one specific reaction. Enzymes are reusable. The same molecule catalyzes thousands of reactions in its lifetime. Three things. 1. Enzymes are proteins, so their shape is everything. 2. Each enzyme is specific to one substrate, like a lock and single key. 3. Enzymes work at body temperature without high heat because they lower the activation energy of the reaction they catalyze. Common slip: enzymes are not alive. They don't die. The proper word is denature, when the shape of the active site is destroyed by heat or extreme pH. And don't say enzymes work on all foods. Each enzyme catalyzes one specific reaction. Choose the wrong one and nothing happens. Quick example: amylase breaks down starch into the sugar maltose. Protease breaks down proteins into amino acids. Lipase breaks down lipids into fatty acids and glycerol. 3 enzymes, 3 substrates, 3 sets of products. Same general idea. Bring the substrate to the active site, break the bonds. Right, if any of this is going to stick beyond this video, drop into phmaths.com. You'll find a 3-4 week GCSE higher revision planner ready to download, plus the mastery workbooks. One for every higher topic, every answer fully worked, every common slip flagged. Simple explanations, serious results. See you next video.